My name is Hayyan Hadi and this is my Abdus Salam science fair project. What agent in soaps prevents bacteria from growing on our hands? Well, the agent is sodium hydroxide, and the concentration changes between 0 to 0.5 in most commercial soaps we use. I want to determine the optimal concentration of sodium hydroxide to prevent bacteria growth. Here are some materials I could use. I can go in depth, but that would take a while, so I'm going to stay on this slide for a little bit so you can read it. That should be enough time. Now time to move on to my hypothesis. My alternative hypothesis. The different concentration of sodium hydroxide have a different amounts of bacteria growth. My opposite hypothesis. All concentrations of sodium hydroxide will have the same amount of bacteria growth. The alternative hypothesis is likely correct. I predict the best concentration of sodium hydroxide will be 0.25, because 0.25 is half of the total, which I think will be the best. Hypothesis reasoning. Survival theory. Survival theory states that all living organisms require nutrients to grow. The nutrient agar in the experiment will act as a medium providing nutrient for the bacteria to grow. In optimal sodium hydroxide concentration chemistry, it is known that a greater concentration of a strong base will kill more bacteria. So as a concentration of sodium chloroxide increases, more bacteria should die until it reaches a minimum. Once the minimum is reached, as concentration of sodium chloroxide increases, bacteria growth will also increase. Moving on to the next slide. Procedure. The first step is to build an incubator. I did this with a box with my dad. You can take a look at the diagram for yourself. Procedure 2, or step 2. Prepare a petri dish. I prepared many petri dishes. Step 3. Agar plates prepared. Plated and bacteria is placed, because remember we need the agar for the bacteria to grow and survive. And so we just did that. Procedure 4. Concentration of sodium chloroxide. Concentration of sodium chloroxide, 0.5. As you can see, you can take a look at our table. Now let's go on to procedure 5. Bacteria plated. So basically what we did is that we plated that bacteria and then we put it in the incubator for 24 hours so the bacteria could grow. And as you can see in this slide. Variables. The independent variable was the concentration of sodium chloroxide in the solution. The dependent variable was the bacteria's growth. Controls. It would take me a while to go in depth on each control, so I made this chart if you would like to go through it yourself. This means that they will react similarly to the sodium hydroxide. Results. Here is a more optimal way for you to see the results. Results interpretive. In ANOVA test, which hypothesis is correct? So it takes my p alpha value, and if it is lower or greater, it determines if my hypothesis is correct. and my hypothesis is correct. Minimal slope of tangent point at minimum growth, as you can see the other graph. Conclusion. The alternative hypothesis is accepted. The concentration of sodium chloride affects bacteria growth. Minimal bacteria growth occurs in when a percent of concentration of sodium chloride is 0.1.0.198. This will conclude my presentation.